Good evening. Good evening. As you probably all know, I'm Dr. Richard Garassi, president of Wagner College, and I want to welcome you to, I believe, what is now the 13th Kaufman Repage lecture. This lectureship started with the generosity of one of our graduates, Louise Kaufman, and her husband, Dr. Peter Kaufman. They wanted back then to establish the fact that Wagner would have a continuous flow of speakers on really cutting-edge issues across the whole spectrum of the curriculum. And we've had this great run of speakers over the years, from Joel Kotkin, who's an urbanist and wrote about the development and loss of the middle class in a lot of cities through gentrification. We've had Rebecca Skloot, who wrote the famous book, The Immortal Life of Henrietta Lacks, which you may know as the woman whose genes were appropriated, cells were appropriated, which led to a lot of cell biology and genetic research. We've had most recently Billy Collins, the poet laureate of the United States, wonderful person. And last year we had Dr. Catherine Monk, who also presented from Columbia University, who looked at the development of the brain of the fetus as affected by toxicity in the mother's brain and stress and so on and so forth. And I'm so pleased that Jeffrey Krause, our provost, I'll introduce in a second, will introduce our speaker, Dr. Martin Picard from Columbia University. With him tonight is Dr. Mary Elizabeth Sutherland, who's also a person who's a senior editor at Nature and Human Behavior, a wonderful journal. She also has her PhD from McGill University. So Jeffrey Krause is going to make the major introduction. Jeffrey is our provost. He's a longtime political scientist at Wagner, for those of you who don't know Jeffrey. He's most notorious because his triplets are freshmen this year at Wagner. His older daughter graduated with distinctive honors from Wagner as well in economics. So Jeffrey's been a tremendous advocate on the faculty for developing our curriculum and developing our students. So without further ado, Jeffrey.
Thank you. Thank you, Richard. Well, of course, I'd like to welcome you as well to this lecture. Our guest speaker this evening is Dr. Martin Picard, who is the principal investigator at Columbia University's Mitochondrial Signaling Lab. Dr. Picard received his Bachelor of Science with honors from McGill University in 2007. He remained at McGill and received his PhD from McGill in 2012, then moved on to the University of Pennsylvania for a postdoc, and since 2015 he has been at Columbia University. The title of his talk this evening is An Energetic View of the Brain-Body Connection. Dr. Picard, the floor is yours.
Thank you for the introduction. Thank you so much for this invitation to visit Wagner. It's my first time here, my second time on Staten Island, which is this beautiful, green area that I never knew about. So it's such a pleasure, and the campus is absolutely beautiful. And we had the little sunset on the way down, just on the way here. So I can't imagine a better way to discover your college. So what we're going to talk about tonight is an energetic view of how the brain and the mind talks to the body. Probably a lot of you have heard about mind-body or brain-body. How is it that what we feel, what we experience, how does that actually get inside the body to change the biology? And there's good evidence, as I'll talk about, that what we experience influences how we age and the risk of disease and so on. So how does that happen? Because there's not a whole lot known about this topic. And the reason why that's important, and I see a lot of people here in scrubs, and a lot of, I think, nursing students and PA students, and which is fantastic. So this is the future of healthcare. And what I'm going to talk about tonight, I think, has some relevance about how we understand individuals, understand what health is, and then ultimately what we can do to promote health and not just try to treat disease once it's a little advanced. So I'll propose that one challenge for 21st century medicine is the following. So to integrate brain-body factors in medicine and to develop new strategies to promote health across the lifespan. And the reason why that's important is because our current medical system is not so attuned to this, and there's a lot of factors that can influence how we age and our physiology and our biology that we don't really understand. And when we don't understand something, it's really hard to change it. So these are two papers to illustrate the point. One of them is in JAMA, which is an important medical journal, and it's called Psychological Stress and Disease. And really what this paper, that was back in 2007, so that's more than 10 years ago now. And this paper made the case that what we know at this point, 10 years ago, is that there's substantial evidence that the stress that people experience, when you say, I feel really stressed out today, that actually influences wound healing. So how the body repairs itself from a cut, for example, is influenced by the amount of stress that we experience. And one of the landmark studies, for example, showed that if you wound people, and they actually did this in medical students. So they said, let's wound medical students when they're, at the beginning of the semester and, they're in pretty good mood and pretty good shape. But then let's wound them again when they're at the end of the semester and during exam period where they're stressed out. And then the students would report, yes, I feel really stressed right now. And then they would measure how quickly the wound heals. And this is something you can assess objectively. And they found that when they were stressed, the wound healed slower. So that was one of the early pieces of evidence that said what maybe some people know intuitively, that the stress that we experience can actually affect the body. And we're probably all affected by stress in different ways, and we all know our bodies, and some people get gut issues and gastrointestinal issues when they're stressed out. Some other people get headaches, some other people get joint pains, and so on. So there's also an individual component to this, but there's that notion that is substantiated by research. That psychological stress has some relevance for disease. The second piece here at the bottom is by Bruce McEwen, who's here at the Rockefeller University. And here he's making the case for how social stressors, so being low in the food chain, if you want, being of low socioeconomic status, or feeling like you're isolated and you don't have anyone to talk about. These are kind of the social stressors and the psychological stressors that has been demonstrated to have also health effects. And Bruce here makes a case that this actually affects the brain and then affects all sorts of organs in the body. And again, we don't really know the pathways, or we don't really know the mechanisms, biological mechanisms, by which mind stuff, subjective experiences, get translated into real biological changes. So just to illustrate this point, this slide here on the power of perception. This is a study that was done in 2002, and they measured the self-perception of aging. So they would interview people. They interviewed 660 adults over 50 years old. So a large group of older women and men, and then they asked them, how do you feel about aging? And they actually had a semi-structured interview to collect those information. Some people said, aging is great because you get so much wiser, and then you accumulate resources, and you can make connections, and there's so much more you can do as you get older. And then some other people said aging really sucks because things just go downhill, everything's bad, and there's no advantage to aging. So this is self-perception of aging. And then they controlled for other things, medical conditions and physical activity, smoking, and they measured some biological factors like cholesterol, blood pressure. And then they asked, does it matter whether people feel that aging is a good thing or whether they feel like aging is a bad thing? Let's follow these people up for 20 plus years and see what happens to them. So what this graph shows here, I'll just step back a little bit. So what this graph shows, on the y-axis here, you have probability of surviving. So at the beginning, 1 is where everyone's alive. So at the beginning of the study, they interview 660 people, everyone's there, everyone's in the study, so that's 100%. And now this is time 0, and then on the x-axis is the time since the interview. And then you can see this is in years, so 5 years, 10 years, 15, 20, and then 22 years. So this is the result. This line here, so as people basically pass away and people die, then the line drops. So you can see how quickly the line drops in people that have low positive self-perception of aging and people that have high positive self-perception of aging. So people who thought 22.6 years ago that aging was a good thing, this is the rate of mortality, the top line here, versus people who thought aging was a bad thing. And then what you can do is draw a line, and this is called median survival, so 50%. So it's the point at which 50% of the population is still alive. And then if you look at the intercept between this line and the 50% line, and then this one here, you get this difference. That difference is 7.6 years. So just the positive self-perception of aging, right? Your subjective opinion of what aging is associated with almost 8-year difference in survival. And then in that same study, they were able to look at other things. So for example, blood pressure and cholesterol, right? Everyone knows, or everyone has heard, that high blood pressure and high blood cholesterol is bad for you, right? In this particular case, that was associated with a 4-year reduction in lifespan. And in comparison, other data sets that looked BMI, or obesity, body mass index, or exercising, that was associated with a 1 to 3 year difference. So just to put in contrast, this subjective experience of the self-perception of aging is associated with a bigger difference in longevity as an end outcome. That's an important one. So the challenge of 21st century medicine, to integrate brain-body factors in medicine to better understand these relationships so that we can do something about it. So there are two main models that have driven biomedical advances. Actually, one main one, and I'll talk about kind of another one that needs to be integrated with this one. So the anatomical model is the model that we're taught in school. That's the model I was taught under in biology and physiology. So you learn about the organs, and then you learn about the systems, and then if you go down, you learn about the cells and the parts of the cells, and then if you go really further down in the cell nucleus, you learn about the genes, and then you learn that this is the basis for the anatomy. And the anatomy is what it's about. And our whole system of medicine is centered around this anatomical model or this anatomical paradigm. So if there's something wrong with your liver, there must be something wrong in your liver cells. If you have a headache, there must be something wrong with your brain. And if you have a heart problem, there must be something wrong with the heart cells, the cardiomyocytes. Well, what if there's something else? What if there's an energetic defect throughout the whole body that can manifest in different organs? This doesn't really fit within the anatomical model. And so this is what I'm going to propose we need to pay more attention to, and that by focusing on this energetic aspect and this energetic dimension of how the body works and how the body responds to stress and how the body heals, we might learn new principles that can be applied in the clinic. So the lecture tonight is going to be separated into these 3 parts. So first we'll talk about how energy keeps us alive, and just the very simple concept of energy. What is that? And how it's central to life. Then we'll talk about mitochondria as a source of energy. Who's heard about mitochondria here? So in our lab at Columbia, we're all mitochondriacs. So we love mitochondria, and I'll tell you a little bit about why we think they're so important. And then the third part is going to be how does stress, psychological stress, affect the brain and the body. And I'll tell you about some work that we've done in the past and then some work that we're currently doing. So let's start by talking about energy. So we can start by asking a very simple question. What distinguishes living organisms? You look at something that's alive and then you look at something that's dead. How do you tell them apart? Just intuitively.
It has cells.
What's that?
It has cells.
It has cells, yes. But what if the cells are dead? Huh? You could look at a dead animal or a dead body. How do you tell apart the living from the dead?
The cell has activity, I guess.
Activity, yes. What's that?
Mitosis.
Some questions, yes.
Consumption of energy.
Consumption of energy, yes. So one very basic, fundamental aspect of life is that there's a flow of energy. So you look at a rock, for example. The rock can get warmer, it can get cooler, but there's no flow of energy through the rock. The rock doesn't sustain energy flow. You look at a smiling, feeling, thinking person, like Gina here, for example. Gina, you can measure, actually, the flow of energy through the body, right? The breath is a very fundamental one. Breathing is so key. A lot of living things breathe. And the reason we breathe is to bring oxygen to the body. And where does oxygen go? To the blood, yes. And then the blood transports oxygen to the cells. And then the oxygen gets in the cells and goes to mitochondria. Exactly. The reason we breathe is to bring oxygen down to the mitochondria. And that sustains the flow of energy in the body. So the flow of energy is central to life. So how does that happen? And what do mitochondria have to do with this? So we all know about photosynthesis and how there would be no life on this planet if there was no photosynthesis. So what happens during photosynthesis is that the energy from the sun is captured in the chloroplast, and then that generates oxygen and then food substrate. So as plants grow, they make starch, or they make carbohydrates, and then animals go and eat. But then also it makes the oxygen that's needed for the mitochondria. And then what do mitochondria generate? In return, they produce CO2, carbon dioxide, and water. And CO2 and water is exactly what the plant needs to grow. So this is a beautiful cycle of life. And mitochondria is essential to close that cycle, to that portion of the cycle in living organisms that are not plants. But why do we need that energy? And one main reason is that we need that energy to interact with the environment. If we want to be able to perceive something, not just the act of seeing something, you need energy in the retina of your eye so that the photon can excite a cell, and then that can create an action potential that then reaches a part of the brain that perceives or processes that visual stimulus. But so all of this is energy dependent. So we need energy to process information. And we're going to talk about stress. So we'll talk about why energy energy in the context of stress. So if we want to be able to respond to a stressor, let's take wound healing, for example. There's a wound that you can cause to med students during their exam period. So here's a wound. Now the body needs to know, what do I do with this? So first it needs to perceive that there is injury. So that the wound would be the stressor in this case. And there could be all sorts of different stressors and stimuli. It could be auditory, visual, metabolic stressors or signals. They're going to be real or imagined. Or real stressors like a wound. But you can also, while giving a public lecture, be imagining that your integrity is threatened. And we actually use this in the lab. It's called public speaking and socio-evaluative threat. So the body responds to this. You imagine that you're in danger, and the heart accelerates, and you start to sweat, and there are stress hormones that are released, and all of that. So this is happening only because there's energy in the body, and the body is able to perceive this. But then it's not just a reaction. It's not just a reflex. There's some information that's contained within you in form of memories, in the form of what genes you have that predispose you to have a certain constitution. And based on your past experiences, you remember what it is to be in the situation, and you adapt accordingly. But so there's information contained, and that's the bottom part of this figure here. So there's information contained in the form of hormones, in the form of DNA, the epigenome, existing proteins, memories of past exposures. In order for outside stressors or outside exposures to interact with existing information in the body, this needs energy. Otherwise, it'd be like a rock. You can throw a rock in front of an audience. You can throw a rock, to a knife. It's not going to do anything. It's not going to respond in an intelligent manner like a living organism would. In large part because there's no flow of energy through the rock. So that's the basic. We need energy to be alive. And then we'll talk a bit more about mitochondria and how to sustain that. So one key point is that mitochondria are the origin of complex life. So we as humans, as multicellular organisms and mammals, would not exist if it was not for mitochondria. So about 1.2 to 1.5 billion years ago, the story goes that the only thing that existed on the planet was unicellular life forms. So little bacteria. There was no plants, no flies, no worms, no mice, no birds, nothing, but unicellular organisms. Some bacteria were not able to use oxygen for their metabolism and to make energy. They were only fermenting bacteria like yeast. But some other bacteria were able to use oxygen. These were aerobic bacteria. And then the story goes that there's a bigger anaerobic bacterium, a bacterium that cannot use oxygen, that engulfed a smaller one that was able to use oxygen, the aerobic bacterium. And then instead of doing what it normally do, which would be to engulf it, digest it, and then use it for food, it sort of kept it around. And that little aerobic bacterium became the mitochondria. So somehow they developed this endosymbiotic relationship, where the big one was deriving something from the small one, and the small aerobic bacterium was enjoying maybe being in the comfort of this protected environment in the bigger bacterium. So the result is today's multicellular life. And there's very good evidence that the cellular life that we enjoy today would not be possible without this endosymbiosis. So if we break down the current model of the cell in two main parts, we can say there's a nucleus. And in the nucleus, there's a genome, and we have chromosomes. And the chromosomes basically contain genes. And the genes code for proteins that provide structure to the organism. And then another very important part of the cell in the cytoplasm are the mitochondria. And what the mitochondria talked about, they take the oxygen that we breathe in, the food substrates that we consume. So most people here probably just had dinner. So we just provided food substrates for mitochondria, and we're all breathing every few seconds. And that goes into the mitochondria to produce energy in every cell of the body. And here's a little illustration of this endosymbiosis. So here's the cell, the nucleus, and the incorporation of the aerobic prokaryote, or the bacteria, that made multicellular life possible. A very important part of this relationship is that it's still a relationship. And as with any relationship, communication is so important. Cannot stress this enough for the young people. So there's very important communication between the mitochondria and the nucleus. And together, they make this life possible. It's a beautiful story. So now, because this is so important, this was so important to making multicellular life possible. We are a big multicellular organism. So it's no wonder that if the mitochondria don't work properly, something is going to go wrong in this big multicellular organism. And this is probably is part of the origin of why mitochondria is being studied to such an extent in biomedical sciences. And as the years go by, there's more and more research being done on mitochondria. So here on this graph, it just illustrates this point. This is the proportion. So what percentage of all of the publications in the scientific community are about different parts of the cell? And you can see there are some, the gray line is about the nucleus, and you have the endoplasmic reticulum, where proteins are synthesized and so on, the Golgi, where things are packaged and shipped out, and then the mitochondria, where energy is produced and signals are generated. And then you can see what happened to the number of the publications as the years went by. This is from 1980 all the way here to 2015. And then this big surge here in the number of publications in the biological sciences was when a technique in the lab was invented called polymerase chain reaction. So we could start to look at DNA. And this spurred molecular biology. And then you see all of these increase, especially the nucleus. So we could start to study DNA. Then you see also a big rise in the mitochondria. Then the other organelles. And then you see as time is going by, other things have happened. You know, the first mitochondrial DNA mutations were identified. So people discovered, wow, the mitochondria which have their genome, and we'll get to this, when there's a mutation in the mitochondria, this actually causes disease. This was the first time we realized that mitochondria not only sustain life, but when they go bad, they can cause disease. And then here in 2001, the genome project, the Human Genome Project was completed. Who's heard of the Human Genome Project? Yes, a lot of people. Huh? What was the goal of the Human Genome Project? What's that? Yes, to map the entire genome with the hope to identify genes that would be the cause of diabetes, find the gene for Alzheimer's, find the gene for Parkinson's, find the gene for heart disease, and so on. But unfortunately, it didn't turn out that way. So you can see up to 2001, the number of publications about the nucleus were increasing, and then by this point, people realized, oh, the answers are not going to be in the genome. And then, interestingly, the number of publications about the nucleus has been on the decline. The number of publications about mitochondria has been on the rise. And the point here is that I think there's this recognition that we need to reorient our emphasis. Here, you know, to study mitochondria, but to understand the energetic dimension of how the system works. So where are mitochondria? They're in every cell of the body, except one cell type. Anybody knows?
I think red blood cells.
Red blood cells, yes. Because what do red blood cells do? They transport oxygen. So if you had mitochondria in the red blood cells, they would consume the oxygen that are destined to the target cells, or to the end cells in your muscle, in your brain, in your heart, and so on. So red blood cells don't have mitochondria because their job is to transport the oxygen. But every other cell in the body has mitochondria. And a very important part about mitochondria is that they're only organelle to have their own genome. So besides the nucleus that has 23 pairs of chromosomes and roughly 25,000 genes, all of the other organelles don't have DNA. It's all in the nucleus, except mitochondria, which are these little circles here. And then you see here this magnified, and that's called the mitochondrial DNA, or mtDNA. And the mitochondrial genome is characterized by a very peculiar way of inheritance. We get half of our genome from mom, and half of the genome from dad, right, in the nucleus. What about the mitochondrial genome?
Just the mother.
Just the mother, exactly. Power to the mother. So the mitochondrial DNA is maternally inherited, and that was discovered in 1981 by Doug Wallace. So we get our mitochondrial DNA from the mother for some reason that is not fully understood, but there's probably some wisdom in there. So what do mitochondria look like? So young audience here, lots of people training in the health professions. When you want to know what something looks like, what do? You're sitting at your computer and you want to know, what does that look like? You Google it, exactly. This is what happens if you Google mitochondria. And the dominant picture here is that mitochondria, or is this bean or peanut shape, or whatever this shape you want to attribute it to. So it's this bean-shaped organelle. And if some of them have this like swimming pool structure in the middle, and there are two membranes, this outer mitochondrial membrane, the inner mitochondrial membrane. These invaginations are called the cristae. This is where the action happens. This is where the oxygen is consumed and the energy is synthesized in those invaginations. But mostly they're beans. This is what you see in textbooks, and maybe things are changing a little bit now, but this is what Google tells you. You've got to believe what Google tells you. But mitochondria are living organisms. And what you see on the page of a textbook or on the page of Google Images, if you go to Google Video, then it might be a different story. I advise people to do this. But what happens if you happen to be in a laboratory and you put living cells under a microscope, then you put something in the cells that make the mitochondria fluorescent. And you can do this quite easily. And then after 30 minutes, you come back and then you take your cells, you put that fluorescent mitochondrial probe, and then you put this under the microscope, then you peek down the eyepieces. What does that look like? So we can take cells, genus cells, for example, then put them under the microscope. This is what it looks like. So this outline here is roughly the outline of that cell. Here in the middle, this big central structure is the cell nucleus. So this is where the genome is. This is where the chromosomes sit. All of these squiggly, dynamic, moving, interacting spaghettis, or the mitochondria. Right? These are the living mitochondria in the cell. If you just bring your attention to this little part, for example, you can see how much movement there is. So mitochondria are living organisms. Maybe some of you can notice mitochondria are all around the nucleus. They're all around the nucleus probably for multiple reasons, but they're there in part to provide energy to regulate the genome, but they also provide input. They provide signals to tell the nuclear genome and to tell the cell, there's not a lot of energy right now, we should really upregulate this gene. Or right now there's way too much energy, we should divide. So mitochondria have a big say in whether the cell divides, whether the cell transforms into something else, or whether the cell dies. Now, the main process of cellular death, apoptosis, is controlled by the mitochondria. So that's how important of a say they have in cellular life and cellular death. Let's look at this other movie. This is also a movie of mitochondria. If you just take a second to look at this. So we can focus our attention, for example, on these little guys here. There are two little mitochondria. You can see they're kind of hanging out together. This long one comes along. Now there are three interacting together, and then this long one says, I've had enough, and then it just goes away. So you can make all sorts of stories about this, but the point is, this looks a lot like if you were looking at an ant colony, or some small, primitive social organelles, or social, insects or whatever. They're interacting, they're exchanging information. We know mitochondria are talking to each other, and you can see this in living cells under the microscope. This is another example of a cell where you make the mitochondria fluorescent, and you ask, what do mitochondria look like? You can do this also in people, and that's something that our lab does. So we want to know, what do mitochondria look like, not in a cell in a dish, but in someone's muscle, for example, or in someone's blood cells. What do mitochondria look like? So we can do this. This here is a piece of muscle. So if you take a muscle biopsy, and maybe you recognize this schematic from your textbook on human anatomy and physiology, you see all of these striated muscles. So every muscle, like the bicep, the quadricep, the deltoid, and all of these muscles, they look like this. Every little fiber here, this long spaghetti structure, this is a muscle fiber. So that's one cell that can contract. And this whole thing here, this is actually a piece of mouse muscle. So not a human muscle, but it looks very similar. So this could be, when I put Gina's muscle, so we can take muscle biopsies. And then what we can do with this, and then look at the mitochondria. These are mitochondria reconstructed in 3 dimensions. So this is the first time we are able to visualize mitochondria, human mitochondria in 3 dimensions. So here we're looking at what mitochondria look like in someone's leg. So this is a person who donated a piece of muscle for a research study, and now we reconstructed the mitochondria in 3 dimensions. Every little color here, you see this large mitochondria here that's turquoise, and you see this one that's purple, and this little one here that's green. So each color is a different mitochondrion. You can see how much differences there are between them. They're not all like beans. They don't all look the same. They're very heterogeneous. So every mitochondria is unique in a way. And we can look at things even more closely. So this is using electron microscopy. And by using electron microscopy, we can not only dive inside the cell to see these mitochondria in 3 dimensions, we can look inside mitochondria. And now we're looking inside mitochondria, which have these squiggly lines where the action happens, I said earlier. So here we're looking at this kind of image. You see that there's a central mitochondrion here. There's another one here on the top right, another one here, another one here, one here, and then one there. And then you see the lines here as we're moving in and out of the mitochondria. So this is what the microscope is doing here. So what maybe you can notice, this central mitochondrion has one part of the same mitochondrion. This is one part that has all of these cristae, these lines, that are nicely parallel aligned. And then there's another part of the same mitochondrion that has the cristae, they're all disorganized. So it looks like there's chaos inside the mitochondria. What's different between the part here on the left and the part here on the right?
One looks uniform and one looks very displastic.
Yeah, so what's different between the uniform part and the disorganized part? One of the differences is that this part here that's disorganized, it's not facing another mitochondrion. It doesn't have a buddy to connect to. All of the other sides of this mitochondrion are touching other mitochondria. So there's something about 2 mitochondria touching each other that is helping to organize the internal structure. That's like social support at the intracellular level. If we also count the number of cristae, and the number of cristae is really important because it gives the mitochondria the ability to make energy. There's also more cristae when the mitochondria are touching each other. You can see here, and if you count the number of cristae compared to here, there's a lot more where there's mito-mito contact. Anything else that people notice? What about if you start to look at between the mitochondria? Yeah. That's probably the most interesting thing in this video. Mitochondria are talking to each other. And the cristae are physically aligned between each other. So you have this central mitochondrion, which is physically separate from the other one, yet the cristae are aligning between them. And you can see this as we go in and out of the mitochondria, the cristae are following each other. and even curving to quite some extent. And in biophysical terms, this is not the most natural conformation. There needs to be some input of energy in order to bend the membrane to reach the border so that it can reach other membranes here. So there's something happening here between the mitochondria that's organizing. So there's communication happening that's organizing the internal structure of the mitochondria. This is the first physical evidence that we've observed, and that was published in the scientific community to show that mitochondria actually talk to each other. Now that we know this is changing a lot of things, because now they're not only little beings that make energy, so a little powerhouse, which is a very mechanical thing. They're living organisms that talk to each other and exchange information. Then that leads to a whole set of new scientific questions. Why do they do this? What are they saying? How are they saying it? And we don't have answers to these questions. But now we can start to look at this in people. This is another kind of structure that we discovered. So it's called nanotunnels. So you can see here, there's one mitochondrion. Here's another one, here's another one, here's another one. So in the classical textbook, you would see these four little things, and then you would say, okay, there's 4 mitochondria. But then here, there are these tubular connections between them. Something else in nature that does this are bacteria. What did we see earlier? Where do mitochondria come from? Come from bacteria. Yeah? So they used to be bacteria, so that making tubular connections to reach another organism, so one mitochondrion creating a tubular connection to reach another organism is an evolutionary conserved thing all the way down from bacteria, and now mitochondria do this. You could even say maybe in this idea of tubular connections, now we know cells can do this as well. That was discovered only in the last 5 years or so. Cells can make these nanotubes, and then they can exchange mitochondria between cells, and they can exchange other kind of organelles. What else in kind of the human world, the human scale, does this? To have a tubular connection to establish a privileged connection between two things. What's up? Yes? Yes, children, yes. So the fetus. The fetus is connected to the mother through a tubular connection. So that's an evolutionary conserved principle. If you want to connect two things, but you want them to remain separate, you don't entirely fuse them, you make them separate, but then you create a tubular connection so that you can regulate the exchanges between them. So that's how we're starting to think about mitochondrial nanotunnels and their purpose, what their purpose might be in biology. So now we've talked about mitochondria and how beautiful they are, how diverse, and how interconnected they are. But now we'll draw a parallel with the human brain, which is also tremendously connected, and which is also highly organized in the same way, or in a similar way that mitochondria are. And then we'll talk about how does stress affect the mitochondria and affect the human organism. So one question that we're interested to address for the greater purpose of creating a new understanding of the factors that influence human health and human disease is, do psychological stress and do psychological factors influence health? And do they influence our mitochondria? So there's a variety of number of stressors. It can be just overwork, can be worry, it can be other kind of negative emotions. It can also be positive emotions. So you have Gina here, you have someone who's grateful and feeling love, and you have someone who's super excited because you just won a contest or a game. All of these things are positive or negative emotions, and we know from experience and from measuring people in the lab, if you hook up electrodes and you look at an ECG, when someone gets excited, the heart rate increases, 20, 30 beats. When someone gets angry, the heart rate and blood pressure also increases. There's a direct, within seconds, a direct connection between emotions and subjective psychological experiences and the body. So now we're asking, could these things also have an effect on mitochondria? Could they change the interconnectivity between the mitochondria, or the ability of mitochondria to make energy? If they do, that would be major, because that would change how energy flows in the body. And if you change how energy flows in the body, you can change the function of organs and systems and how a person actually perceives and is able to respond to a stressor. So if we take stress here for a second, so we can consider that there is a stress to mitochondria to disease axis. So that would be, you experience stress, then it stresses out your mitochondria, And when your mitochondria are stressed out, then they actually cause changes in the cells that can promote disease. So this is a stressed mitochondria to disease axis. We can also think about it in a more positive way, which would be, the mind or any mind psychological experiences, and then to changing the mitochondria and then promoting health, because that might very well happen. We tend to be more focused on the disease and the pathology in medical training and practice, but there's this other dimension that if we could learn how to tap into, that would be fantastic. So this is the basic model. So in science, if you want to test something that's a very large idea like this one, you need to establish a very simple and ideally linear model to the extent that this can accurately reflect the reality. So here we have a model where we have stressors, and then we say we have an arrow that says stress pathophysiology. So this is causing disease-associated changes in target organs, including the brain and other organs. And then we're asking with these arrows, is the mitochondria affected by the stressors, and then can the mitochondria cause a stress pathophysiology? So these are kind of two elements in this causal model. If stress can change the mitochondria, and then the mitochondria can cause stress pathophysiology, then this satisfies the conditions for mitochondria to be a mediator. In other way, they would be the portal between the experience of the stress and the change in the body. So let's look at this first. Is there evidence that psychological stress or psychological factors could influence the mitochondria? So to do this, we did a little study in collaboration with Alyssa Eppel at UCSF. And so we asked people these questions. This is just a few examples. So we asked them, for each of the emotions listed below, please tell us how much you felt this emotion this evening. So this is a questionnaire to complete in the evening, and they also had a similar questionnaire to complete in the morning. So in the evening, imagine you just come back from school or from work, and you're at home, and it's like 8, 9, 10 p.m., and then before you go to bed, you answer this question. So what's the most amused, fun-loving, silly you felt? So you get a sense for what that feels like. And maybe you say, not at all, 0, or you say extremely, that's how I felt all day. And then there are all of these other questions. Here's another positive one. Grateful, appreciative, and thankful. And then there are also these negative ones, or negatively coded, I should say. Angry, irritated, annoyed, or disgust, distaste, revulsion, guilty, repentant, blameworthy, and so on. So you get a score for each of these questions. So then at the end, you can have a number. How high the number is, how high is the score for the positive emotions, how high is the score for the negative emotions. So then you start to get a sense, how is the person feeling this evening? So once you have that, then you can ask, well, is that correlated statistically? So you can do a statistical test to say, is that correlated to that person's mitochondria? So then we had these people come in, they gave blood, then from the white blood cells, not the red blood cells, right, which don't have mitochondria, the white blood cells, the immune cells, they have mitochondria. We extract them, and then we measure the mitochondrial health. So we measure mitochondrial energy production capacity. And then we ask how they felt the day before, so the night before they came in. Is that correlated to their mitochondrial health this morning? So this is the result. Basically, mood, or these positive, negative psychological state the day after, predicted mitochondrial function the day after. The day before, sorry, predicted the day after. So here we have this graph that shows mitochondrial health. So this is this mitochondrial health index that we've developed. And this is in arbitrary units. So you can see that and the two classifications, the two colors is negative mood and positive mood. And then we separated people in tertiles. So low is the lowest third, and then moderate is the middle third, and then high is the top third. So here there was about 90 people, so imagine there's 30 people in each category. So in the mild, there's 30 people, let's say for the positive mood, the 30 people that scored the lowest on the positive, funny, fun-loving, silly, kind of questions. And then you can see these people that say they don't feel very positive, they're also those that have the lowest mitochondrial health index. And then people that say they felt very positive, they're those that have the highest mitochondrial health index. And as expected, you see for the negative emotions, people who felt the least negative, who scored very low on the negative questions, they had the highest mitochondrial health. People who said, I feel very negative tonight, then they had the lowest mitochondrial health. So to see this going in two different directions, negative mood, more negative mood, lower mitochondrial health, more positive mood, better mitochondrial health, this is making us more confident that there's a real association between how people felt, their psychological states, and the mitochondria. And if you break this down, the bottom line is that mood, in this case, predict 10 to 15% of the mitochondrial health the day after. This is a sizable fraction. And there's a lot of things like smoking or cholesterol or blood pressure. We talked about effect sizes earlier, you know, how much change it makes in longevity. 10-15% change in the biological function just with changing psychological state is a big effect. So now we're in the process of doing more studies to understand this. So if we go back to this little model, now I've showed you some data that shows psychological stress potentially being affected with influencing mitochondrial health. Now what about the other way around? If we change our mitochondria, do we change how the body functions and how the body perceives stress? So let's focus on the brain for a moment. This is a human brain. It's very cool. Who's seen a brain like this before? So this is from an MRI, magnetic resonance imaging of a young female. And you can see as we go through the brain that there are these squiggles also. You could say maybe reminiscent of the crystal in the mitochondria. But these different parts of the brain are also interconnected between one another. And you can do a certain type of imaging that lets you see the communication path between different parts of the brain. That's called tractography. So you can look at the different tracks between the brain. This is what it looks like in a different person, actually. This is a 40-year-old male. And then each little fiber here, each little spaghetti, is a bundle of axons from neurons. And then you can see how they go from, for example, this front part of the brain, and then all the way back to the back of the brain, which is the part where we process visual information. So this lets you see in 3 dimensions. We can rotate this and look at, quantify how many fibers are going this way, that way, how many fibers are going from one side to the other, and so on. So this allows you to see the connectivity, just like we look at the connectivity between the mitochondria, but here between the different parts of the brain. So we're running a study now. It's called the MISB study. It stands for Mitochondrial Stress, Brain Imaging, and Epigenetic Study, or MISBID. You can see the little brain here, the little DNA strand around the eye. These are epigenetic marks, so I'm not going to get into epigenetics, but for those of you who are interested, it's a layer of regulation on top of the human genome, which can be affected by stress, among other things. So in the MISB study, we're trying to understand this exact relationship. How does mitochondrial function regulate biomarkers of aging, biomarkers of disease? How does it regulate the body and how it responds to stress? And then whether brain activity actually mediates that process. So we're recruiting people, like Gina, for example, and they come into the lab, and then we take some blood from Gina, and then we profile the mitochondrial health. So we try to ask questions to mitochondria, we stress them, we probe them in different ways, the same way we would with people, and like kind of like a diagnostic interview. So we have ways in the laboratory to test them in different ways and see how they respond. And then once we've established the mitochondrial phenotype, or the type of mitochondria, then we measure biomarkers from the blood, and then we look at how the brain functions. We put our participants in the scanner. And then this is the first study to look at the relationship between cellular energetics and mitochondria, and how the brain works, and then how the body responds to stressors. So we're very excited about that study. Our whole lab and a nice team of collaborators is helping to make this possible. And hopefully we're going to learn something meaningful that will help us to draw bigger, to learn new things and have better approaches to promoting people's resilience. Another thing that I just want to touch on with this one slide is something we're interested to understand is aging. How do you study aging if you want to understand what influences lifespan? Because we know that psychological stress can accelerate the aging process. How does that happen at the cellular level? We don't know that. And then if it's true, what can we do to mitigate this and to actually help people age more in a healthier way? So it's really hard to study lifespan because people live really long. So as a researcher, how do you study lifespan? So one way that we've found that we can study aging is that we can take a little piece of skin, the same way that you do to medical students to study their wound healing. So you take that little piece of skin, you put it in a dish, and then you can actually monitor the cells. And we found that when the cells are taken out of the body, they behave, they continue to age in a similar way that they do in the body, but at a much faster rate. So they age almost 100 times faster in the dish than when they're in the body. So then that allows us, for example, to take genus cells, put them in a dish like this. So you can see the cells here that when they're young, when they're middle age, so that would be like 50s, and then in their 80s, you see the cells kind of look different. And then something that takes 80 years of person life to accomplish takes about 6 months in the dish. So there's a student in the lab, Gabriel, who's doing these kind of studies, and then he's taking cells from different people, women and men, putting them in a dish, and then looking at their aging trajectories. And then he's stressing some of them with stress hormones, the same kind of stress hormones that are released when we're exposed to psychological stress. And then he's asking, does that change how these cells age? And by doing this, we're learning some interesting things. This is a graph that shows how many times the cells divide, and how many times a cell divide is representative of the lifespan of the cell. So the more divisions a cell can do means the longer it lives. And then here on the x-axis you have the time. So as the time passes, you see the cells get older and older, and then at some point they plateau, and then that's end of life. In red here is when we grow the cells with stress hormones. So they get older much faster, and they reach end of life much faster as well. So those are the kind of results that we're just starting to collect and to study to understand the relationship between here stress hormones and cellular aging. And we've also found in a very interesting way that graying of hair is actually reversible, at least in a temporary way. So if you study people that are in this critical phase where they start to have white hairs along the temples, and then it starts to spread. So these people, you can find hairs that are dark and they turn white. So you can actually see that transition. You can say, this hair just became old. But you can also see this hair that goes back to being dark. So it's rejuvenated in a way. So then we found this now in about 12 people, and it's very interesting because it happens in people of different ages, and it seems like it might be associated with psychological states. So that's something we're actively studying, but I don't have more to report on this. Maybe next year or in a few years. I want to point out, as we get closer to the end, to study these things, we need to take a lifespan approach. As Catherine Monk discussed last year, and as is becoming increasingly apparent, aging trajectories, but also health profiles are determined very early on. As the baby is still in utero, being exposed through the mother to different things. As a child, the environment that you're that you're exposed to as you grow and develop is also critical to shape how the brain is wired and how you interact with your environment. And then as you get older, also things change in the body. And the treatments that are developed in middle-aged people or in young adults, like in clinical trials, might not be the best treatments that we can apply for kids or for older people. So thinking about medical treatments or these relationships between mind and mitochondria and mind and body might also be different between across a lifespan. So if we go back to this pathway, what I've showed you is some evidence that psychological stress can change the mitochondria, and the mitochondria can also change different aspects of how the body works. So if we summarize this in a way that's as simple and intelligible, so we try to understand how experiences, daily experiences, and what we feel, how that gets transduced into the biology. And we can call this biological embedding of light exposures. So how does that happen? So our model is that first there needs to be something that senses the stress, that perceives the outside signal. So that's sensing. And then that signal needs to reach whatever receptor there is. And our model and what the data shows is that mitochondria might be that sensing. And this needs energy. You need energy in order to sense that external signal and to just perceive the stress. And then the body needs to respond to that. So in order to do this, then the mitochondria need to integrate that information, and then they produce another signal that eventually leads to the imprint or the transduction of that subjective feeling into something real biological. The mitochondria have the ability to do those three things: to sense, to integrate, and then to produce signals. So if we don't have properly functioning mitochondria, then the experience of the stress cannot be transduced. But with the mitochondria, then that should be possible. So I want to go back to this in closing. The challenge for 21st century medicine is to integrate those domains and those aspects of human health, and that links mind and the body. And that's something that is hard to integrate for many reasons, and it's not typically part of medical training nor practice, in part because we don't have an understanding of the mechanisms by which this happens. And understanding the energetic processes and mechanisms for this might help to do that. But also it calls for a more integrative approach to seeing the body not just as a machine, as a powerhouse, so on, but as an integrated organism that lives in the context of an environment where social interactions and psychological factors are meaningful, not only to mental health, but to physical health. And what I showed you tonight is that understanding more about how energy interacts with information and how that allows us to interact with the environment and to others around us might help us to reach that goal. And I would propose that this is going to help three main things. First, to integrate knowledge of brain-body processes, what we learn about how the brain and the body communicate and interact. If we can understand that, for example, the placebo effect, if we can understand how the placebo effect works, and where you can subject someone to something which changes directly something in the body. We have no idea how that works, but if we could, and we could harness that, we could promote health probably in a very powerful way. Eventually, also, we need to capture the complexity of diseases and individuals. So then Hippocrates said something at some point that it is much more important to understand which person has a disease than what disease a person has. And that wise statement, I think, talks to the fact that not everyone might be helped by the same treatment, even if they have the same quote-unquote diagnosis. Everyone's different, and everyone's same disease can also be different. And by capturing the complexity of the diseases, but also the individuals, not only at the anatomical level, but at the energetic level, that might bring us some powerful tools to do this. And then finally, these things combined could help us to develop a holistic approach, not only to prevent disease and to treat disease, but also to promote health. So I want to end by thanking our wonderful team. This kind of work is done with a group of people that feel passionate and care about these kind of issues. And this is our team. Our lab is here in this building. So we're up on the island by the river. We have a bit of green, not as much as Wagner, but we have a bit. And this is our team here on Long Beach this summer, and they're a wonderful team of people in the lab and who interact with the patients and the participants. These are wonderful collaborators without whom this work would not be possible. Particularly the MISB study is conducted by this fantastic group of people here, where we have Bruce McEwen, who's an expert of brain and stress adaptation, Michio Hirano, who's a clinician who sees patients with mitochondrial disease. So we're actually working with individuals that have genetic defect in their mitochondria, and we're asking, how do they perceive stress? How do they perceive the world if their mitochondria are not functioning properly? And we have two wonderful neuroimaging experts who look at how the brain talks to each other, how different parts of the brain are connected functionally, but also in the connectivity. Richard Sloan, who's an expert in stress reactivity, and our biostatistics experts, and a lot of other colleagues. And of course, the funding sources that make this work possible, which is very important. So if you have any questions, I think there's some time.
Dr. Picard, thank you for that wonderful talk. I know I speak for myself and some other folks in this room in encouraging you to move forward on that research on the reversal of gray hair. I think that's of great interest to some of us here. So we do have some time. Any questions? Right over there. Yes?
So I have two questions. One is, have you studied the mitochondria of individuals with different disease processes? Like I'm thinking schizophrenia versus the healthy individual. And are those mitochondria corresponding, or as you said, communicating with each other differently?
Yes. So in different disease conditions, are the mitochondria miscommunicating? That's a great question. And the answer is we've started to do this, but there's a lot more work that needs to be done. We've never looked in schizophrenia or in other psychiatric conditions. That would be very interesting to do. At the moment, we're studying individuals with mitochondrial disease, and we're looking at how psychological states are linked with mitochondria in this population, but there's definitely a lot more work to be done in that area.
Right. You had a second question, Nora?
Yes.
Okay.
In the same vein, have you looked at the mitochondria of different cells? So I'm thinking cardiac cells versus neurons.
Yes.
And are they corresponding differently?
Yes. Fantastic question. Are all mitochondria created equal? So the mitochondria in maybe that'll be better for people. Is that better? Is that better?
Yeah.
So are the mitochondria in the heart different than the mitochondria in the brain? And are they different than those in the liver and in the skin and so on? The answer is yes. And mitochondria, the same way that different cells are different, the mitochondria in different cell types are also quite different. And are they affected by stress differently? We don't know. But likely they are. The same way that different people are affected by stressors to different extent, that different cell types function and are more vulnerable or resilient to certain stressors. We imagine mitochondria are going to show different vulnerabilities or resiliencies to different stressors. That's a fantastic question.
Gentleman in the white shirt had a question right there. So we'll get the mic back to you.
Me at home. Let's flip it. You said that the female is the sole generator of the mitochondria. Does that mean that it's constant over time?
Can you clarify that it's constant?
If you don't require two to change, only one, does that mean that the mitochondria from the female will be constant in perpetuity?
Fantastic question. Yes. So people that actually use that principle, that mitochondrial DNA is inherited from the mother down to their offspring, both the males and the females, but then it's only the females that pass it on. And that's how we know that we come from Africa. You know, that we're all, we all originated from Africa, 100,000+ years ago, from looking at the maternal lineage. So someone, actually Doug Wallace and his team, back in the 80s and 90s, they went around the world to Aboriginal populations that had not mixed and not, flying around and traveled. And they looked at their mitochondrial DNA, and they were able to actually trace maternal lineages the whole way from now to modern populations all the way to Aboriginal populations. So it is constant. And actually in forensic medicine, that's how people are recognized. If you find hairs on a crime scene, it's the mitochondrial DNA that is looked at to identify people.
So it does not evolve?
With time, it acquires mutations. And that's something I did not touch on, but with time, across someone's lifespan. So as we age, mitochondrial DNA accumulates mutations. Some people think that's why we age, actually. If we think about it fundamentally, we don't know why we age. What in the body keeps track of time? Where's the clock? We don't know. Some people think that it's the mitochondria as a clock, and what actually records the passage of time are the mutations in the mitochondrial DNA. There are experiments that have been done in animal models and so on that suggest that this is the case. And,
When you pass that gene on, does it go to the mutant gene or goes to the primary gene?
So if a woman, for example, acquires a new mutation, that can be passed on then to the subsequent generation. Yeah.
I'll get a student question right over here.
Yes. This is probably,
Why don't you stand up? That might be easier.
okay, in the microphone? Okay. Okay, this is probably less of a question and more of a comment, but since you mentioned the relationship between mitochondria and human diseases, maybe there's probably a relationship to how viruses, bacteria, and fungi might affect mitochondria.
Mm-hmm. Yes. Very nice. So the immune system actually needs the mitochondria to recognize pathogens. And there was a beautiful study that was done a few years ago where they took immune cells, they exposed these cells to bacteria, and then they asked, how do the cells respond? And then you see the immune cells, they perceive the bacteria, and then boom, they mount an immune response, and they produce what's called cytokines, inflammatory molecules, that then goes and alert the rest of the organism. Then what they did is they said, okay, let's knock down the mitochondria, and then expose them to the pathogen again. So if you knock down the mitochondria, you basically decouple their membrane. That's a technical term, but it basically means you de-energize the mitochondria, and then you expose them to the same pathogen, then the cells, no response. So the mitochondria are involved in recognizing pathogens from that perspective. It's a great question.
Okay. Warren Prochon, Chair of our board.
I usually don't need a microphone, but
Okay.
really enjoyed your presentation a great deal, and I am going to get online for reversibility of gray hair. Two questions. Let me make a comment before a question. You were talking about genome mapping. If anybody here is a New York Times reader, in the science section yesterday, somebody was talking about genetic mapping for how to give blue-eyed babies, which I think to a lot of people would seem a perversion of the purpose of genome mapping. Anyway, just move on. I'd be interested in your comment about that. I'm a psychiatrist. We in psychiatry obviously think of, quote, psychological stress as being every bit as harmful as physical stresses. And it seems to me, even though you've mentioned you haven't specifically studied psychiatric illnesses yet, it seems to me you're very much leaving open that possibility. You're talking about stress, I think, in a broad sense that could include psychological, physical, etc. And I would assume that at the fundamental cellular level, the phenomena may be similar. Curious about how it gets from the macro level of various biological systems down to the cellular level, and what kind of thoughts you have about what those steps might be. I'm sure they're very complicated. And then a second kind of simpler question, a little whimsical. You were talking earlier about the damaging effect of isolation, and I would certainly see that in terms of a psychiatric patient population. But when you showed what's going on in the mitochondria, it looked like those tubules that were isolated from others had a less salutary fate, less salubrious fate than those that made connections. So I wonder if in any way you see maybe some sort of similarity between the effect of isolation at the cellular level and the macro level.
Yes. So are there conserved principles at the human social level and the intracellular level? Yeah. That's a beautiful, largely unexplored question. And we do see this kind of behavior in mitochondria. So some mitochondria do become excluded from the cell, from the rest of the mitochondrial network. And in many cases, that is the first necessary step for the mitochondria then to become degraded and actually die. So mitochondria have a life cycle. There's another evidence that they're dynamic living organisms. And through this life cycle, at the end of life, they segregate and then they're outside of the network and then they become degraded. So that does happen. Whether certain pathological states are associated with more of this, it seems like it's the case. So in some cases of disease, the mitochondrial networks actually become fragmented. So the tubules disappear, and then one longer mitochondrial tubule can undergo what's called fission, mitochondrial fission. And then in the fission, you get the pinching of a tubule into smaller little units. So that's called, then that isolates mitochondria from one another in a way. If we prevent mitochondria from refusing and reconnecting with one another, then the cell does really bad, and that's actually the first step to cell death. Regarding the other points, we're definitely thinking about psychiatric disorders, and there are reasons to believe that there are mitochondrial impairments in many of those disorders, and actually in the patients that we study with mitochondrial disease. So these are individuals that have primary mitochondrial dysfunction. So they have a mutation in the mitochondrial DNA that prevents the energizing of mitochondria. The incidence of depressive disorder, of anxiety disorder, and potentially of other psychotic disorder is probably much higher. It hasn't been looked at very much because neurologists don't talk a lot to psychiatrists, which is kind of an academic debate. But there's definitely evidence that there's something also going array in the mind of individuals with mitochondrial dysfunction, which is fascinating.
Have a question right back up there. Young lady in the aisle, you had your hand up before?
We have here over here.
Right over here? Okay. Yeah, okay. Let's get one of the students.
I was wondering,
Green light. We have mitochondrial disaster right in front of us.
The microphone is not energized. This one works.
Might be easier if you stand up when you ask your question.
I was wondering, it's fantastic what I saw, what you show us. However, there's certain parts about humanity that would probably be very, very helpful if your expertise and your studies come up with some solutions. For example, as we age, things change in our body, in our mind too. And I was told that Alzheimer's is one of those problems that happens to people, some people, not all people. And I wonder how this relates to your studies, how this could help to find the solution to that problem.
Yes. Yes, great question. So things change as we age, and we could translate that by saying, you know, there are different aging trajectories. Some people age gracefully and remain active and well in terms of psychological well-being until late 80s, 90s, 100s, right? And that's one trajectory. And then there's some other trajectories that are more bumpy. Why is that? And we don't have answers to those questions. And you know, some people are looking for answers for those, you know, that question and other similar questions in the genes. So you can sequence a whole genome and then look for, what's happening now with precision medicine. We're trying to understand the genetic determinants of these kind of aging trajectories and disease susceptibility. There doesn't seem to be a lot of answers, and the best estimates are that 10 to 20% of how long someone lives is determined genetically. 10 to 20% of longevity, which is abysmal in relation to what most people think the genetic contribution is. So that means there's 80 to 90% of your aging trajectory that is shaped by what you eat, your behavior, what you do, what you don't do, how you feel on a daily basis, whether you engage in something meaningful and purposeful. These are huge factors that, you know, remain mostly untapped from a research perspective and from a health promotion and disease prevention perspective. So there's tremendous amount of work to be done there. We have no clue why people get Alzheimer's disease, and hopefully we'll find out. And maybe mitochondria will give us some clues into that.
Right up there. Young lady in the white jacket.
Great presentation, first of all. I had a question. I recently read an article on allostatic load or like chronic stress as a predictor for preterm birth. And they use things such as like cortisol levels and blood pressures to make that relation. Do you think there's something on the mitochondrial level, like biomarkers, like biological biomarkers, where we can look at mitochondrial stress?
Yes. So we're trying to develop biomarkers like this. And allostatic load was developed by Bruce McEwen. Thank you. Bruce McEwen at Rockefeller University that I mentioned, he developed this concept of allostatic load, which is a beautiful example of what we need more of in medicine and in research. So he said the whole body is an integrated system. You know, there's the brain, and then the brain makes hormones, and then it talks to other organs in the body, like the adrenal glands, which make cortisol, and then that talks to the liver, which will make things like leptin and regulate cholesterol and glucose, and then that goes to the heart, and then it regulates blood pressure, and so on. So everything is interconnected. And so by recognizing this, he came up with this index. He said, if we want to capture how physiologically stressed or in what shape someone is in terms of physiological regulation or dysregulation, we can't measure one thing. You know, we need to integrate multiple things together. And clinically, you would never, you know, assess someone based on one question. There's so many things you want to look at, and so many questions you need to ask, and blood tests you want to do to get a full picture. But reductionism is so beautiful because it's simplistic, and it helps us make progress in many different areas. But it's super limited in the area of human health because things are not simple and things are not linear, and things cannot always be reduced to simple things. So allostatic load, this concept of multi-systemic dysregulation, is beautiful. And with Bruce, we've developed something called mitochondrial allostatic load, or MAL. For the French speaking, mal is it's the bad within. The mitochondrial allostatic load, that's the same concept. The change in shape in mitochondria is not a bad thing, but then if it's chronically in that state, and then that leads to more reactive oxygen species or oxidative stress, and then that leads to more of this and less of that, chronically this can be bad. So we're working now on developing mitochondrial indices, so mitochondrial health index, that are integrative measures that can be more informative than single biomarkers. So I'm not that confident that we're going to come up with one new biomarker that's going to be in all of the clinics, within a few years. But I do think we'll evolve in our understanding and in our ability to integrate knowledge and integrate different kind of measures that can be clinically meaningful and that can help us in research.
Let's see. Right over here.
Could I just say it? I don't.
Yeah, go ahead.
So, cancer cells, they have uncontrolled growth. And since mitochondria are responsible for providing the energy needed for cellular growth, are there differences between non-cancer cells and cancer cells at the mitochondrial level? And if there's any way we could control communication between mitochondria and the nucleus, is there a way we could stop the growth within cancer cells and prevent metastasizing?
That is great. We've thought about that a lot. And what we know is that cancer cell mitochondria are very different. You can basically tell apart a cancer cell from a non-cancer cell just by looking at the metabolism and by looking at the mitochondria. So they're definitely very different. What seems to happen in cancer cells is that they ditch their mitochondria. And that might be one determinant of what allows them to become cancerous in the first place. If we go back to how complex multicellular life happens, that necessarily involved unicellular organism whose sole purpose is to grow and divide. That's what a cancer cell is, pretty much. So you have these unicellular organisms, and then all of a sudden the mitochondria comes along inside the cytoplasm, and then it allows multicellular life to happen. To do this, you need to tame that, you could say, cancerous instinct to just grow and divide and eat food and so on. So the whole organism survives because cells are aware of other cells, and they have this kind of, you could say, consciousness of being in the organism that prevents the growth, prevents metastasis, and so on. So it's possible that for a normal cell that is, for example, in the liver, it knows it's a liver cell, it's surrounded by other liver cells, and it's kind of behaving in a community-oriented way, for that cell to come out of this state of this kind of good citizen, it needs perhaps to ditch its mitochondria. And then by losing touch with their mitochondria, the cell can enter or revert, you could say, revert back to this primitive mode of being that it used to be without mitochondria, which is this more fermentive, different metabolism and just focus on growth and replication and invasion. So that's a very intense area of research at the moment.
We have time for one more question. Let's try to get another student question. Right back there, your hand's up.
This was awesome, but I don't think I need microphones. But this was awesome. My mind is like exploding. But you said that the mitochondria recognizes disease. So have you ever looked at autoimmune diseases and if they're like the mitochondria and those cells are different?
Yes, so you just tap into one of my favorite areas at the moment, and one thing that we're working on very actively. If we think about where mitochondria come from, they used to be bacteria, and what do the immune system recognize? Bacteria and pathogens. So if there are pieces of mitochondria that are released out into the blood this actually triggers the immune system. So mitochondrial DNA, for example, is a very potent immunogen. So if you inject mitochondrial DNA into mice, and these experiments have been done, you get a massive immune response. And if you look in arthritis in the joint and the synovial fluid, there's a lot of inflammation, and there's also, it turns out, a lot of mitochondrial DNA. So some people think that when the mitochondria are unhealthy or they're stressed, however that's operationalized, when the mitochondria are stressed, they can release mitochondrial DNA outside the cell, and then that can trigger immune responses. So that's one potential mechanism by which mitochondria can trigger the immune response that could be involved in autoimmune diseases. So I think autoimmune diseases are still mostly not fully understood, but that's one potential mechanism.
This has been fantastic.
Thank you.